And we have back with us again for a repeat appearance, Fred Brown, our infectious disease expert. And he has been scaring the bejesus out of us now for about 2 months. But but he's saying things that I'm not hearing other people saying, which we really appreciate it.
Yeah.
I know that you were showing me some of the slides you want to introduce at some point here. But really the issue is, I guess, how to test employees when they come back to work, or at least maybe even at the door before they walk through the door to come back to work. Because the last thing you want is somebody to come in and infect everybody, and then you have to shut down, right?
Yeah, that's right. It turns out the way this the virus spreads, it's got a very low what they call K value, and K value means clustering. And unfortunately, it means that one person. We talk about replication of a virus, and that means how much it replicates when one person has it and gives it to the next person, and the next person. And we talk about that pretty simply as well. It's probably around 2.5 to 3 for this in this case. And that means that one person infects 2.5 to 3 people when they get the disease. And that means that, of course, the virus spreads. And what we're trying to do is get the R value below 1. Now, there's another factor that we haven't really talked about called K value. It's the clustering value. And that means that actually 1 to 3.5 doesn't really work in real life. What turns out to be the case when you get very low, in this case, we think it's about a 0.1 K value, which is very low. It means that one person tends to spread the disease a lot. So you have a lot of infected people who don't spread the disease at all, and then one person gives it to 100 people or 200 people, or one location or one event will give it to 100 or 200 people, and that'll cause the overall value to be R2.5. But the real factor was that one or two events, or the 1 or 2 persons, the 1 or 2 locations that really drove the numbers high.
So, Fred, why are some people super spreaders and some people are not? Why is that the case?
Boy, that is a $1 billion question. I'll tell you that. We're trying to figure it out, and one of the ways we do that is with this contact tracing. So after you get the test, you then trace to see who else you've talked to, and then you can find out whether you're a super spreader or whether you're a location you were at was a super spreading location, or whether the activity you did was a super spreading activity. And we're starting to find that some activities are much more likely to spread disease than others. And unfortunately, in rioting, when you're and protesting, I shouldn't say rioting. When we're protesting, we're quite close together, and we're shouting, which is what you want to do, and make your voice heard. And of course, that really tends to spread the virus a lot. So I'd encourage people really to wear masks as much as you can if you're going to be out and about, because there's going to be quite a bit of potentially virus in the air. Every breath you take, it gives out about 3,000 bits of virus particles if you're infected, and if you're shouting or singing or active, it can give as much as 20,000 bits of virus a breath, and all you need is about 1,000 bits to get infected. So you can get a sense of the activity level that you're doing. It's human to human is mostly why this spreads, and these super spreaders, super activities are the reason that we're so worried. And so work activities, obviously, as an opportunity for super spreading. You might become a super spreader location, or the activities you're doing at work might become a super spreading activity, or you might be unfortunate enough to be in a super spreader environment. And so what we're doing now is we're starting to test people and then trace them to understand what's causing the super spreading.
So the auto industry is slowly reopening, but I think they've taken a lot of safeguards from what I've heard from various people, with plastic shields between stations, people wearing masks, people wearing gloves. But we won't really know. I mean, I think it's this week when they're really starting to gear back up, or maybe it was last week. But it'll take a couple of weeks for us to see how well that's really working, right?
Yeah, that's right. And I have worked with a few manufacturing plants to try to reduce, especially early days when we were really concerned about keeping these essential plants working. So one of the plants that I was working with actually was producing large ventilating equipment, which was very critical for keeping people alive. And they really wanted to know what the best practices were. And we developed a few good practices. And I'm happy to take through some of the steps that we did in order to protect the workers there.
Yeah, go right ahead.
We had about 300 workers. This is in Rhode Island and Massachusetts area. And we were actually trying to expand because we needed more ventilators, not less. We were actually expanding at the time. And we had initially one shift. We had all the executives, all the engineers, all the plant workers at the plant. And the first thing we did was we talked to the executives and the engineers and said, do you really need to be at the plant? And they said, actually, we don't actually have to be in the plant all the time. In fact, we don't have to be at the plant much at all frequently. There are a few of them had to be on the plant, but a lot of them didn't. And so we actually got about 40% more plant space out of all those old offices that were then vacated, and they're still vacated today. The executives are all off-site working, engineers still off-site working. They come in every once in a while if they absolutely have to, but those desk spaces, those desks, it became actually part of the manufacturing operation. We were able to get about 40% more plant space out of that than was originally planned, which was great. The other thing we did was we took a look quickly at the workers. Now, if you're talking about young, healthy workers, they're not at a much lower risk than us older guys are. And unfortunately, in this case, we had a lot of older expert people assembling these devices, and so we tried to figure out how we could place them either get them off-site, encourage them to work off-site, see if we could give them different kinds of roles. And have them train, the new people to come in, or to see if we couldn't put them in a more remote part of the environment so they are better protected. And so that's the other thing we did, is as far as trying to get more space, trying to keep the people separated from people who could work off-site and people who had to work on-site. Now, with the people who had to work on-site, we had to train them on new hygiene. And so hopefully, if you're working at a place, and they'll be sending out new standard operating procedures and new hygiene rules. And the first thing we wanted to do was make sure that before you enter the plant, you actually were tested with a thermals test to make sure you didn't have a temperature. That was 88% of the cases, people who have temperatures associated with COVID. It's a symptom of COVID, and 88% of the cases. So we looked at that. And we do this every day for every shift. Now, if you think about it, getting 150 people spaced 6 feet apart and getting them through a 30-second to a minute thermal scan takes time. Mm-hmm. And so we had to stagger the shifts, and we started having guys come in at staggered shift levels, and it took us about an hour and a half to load up the whole plant. So you want to have enough time so you don't have a mad crush at the door. Everyone trying to get in at 7:30 so they can get out at 4:30 or whatever the shift schedules are. So we tried to ramp that out so that we could start at the plant and keep it going, and then we should shut it down at the end of the day to do a deep cleaning at the end of the day. We went from one shift to 2 shifts because we actually doubled our capacity, not because not because we needed the extra space. We had enough space, actually, it turns out. The spacing is sort of interesting. If you do the math, if you're absolutely perfect, you can do 6-foot spacing in a normal room but it takes away about 52 with about 52% of the space. So it takes away 48% of your space if you're doing it right. That's a lot of space to lose, so you have to really think through how much distancing you want to do, how dangerous it really is, and so on, pretty carefully. We checked all the ventilation. We made sure HEPA filters were being removed and changed often, but we still have quite a bit of airflow in the production area when we can. The problem is we're assembling chips and things like that don't really like a lot of airflow. So we had to balance that carefully. But I think we're doing a pretty good job there. So that was the first thing, was just figuring out how to get the shift staggered and how to get people into the building safely. And they have to be tested before they enter the building. So we encourage, we had a parking lot, luckily, and we encouraged workers to come at a certain time with an app, and they would come at their appointed time, and they'd be able to get right through without waiting in line by waiting in their cars. So that's what we did, a lot of work in the cars. We also shut down the cafeteria. We had a nice cafeteria. And we decided, let's let everyone take their breaks and do the cafeteria work in their cars. So we're doing that in the cars. It's not that popular, but it does mean you're keeping that distance right, because our canteen was really tight in that particular case. And so we decided we would bring the food in for all the workers, which is nice, so they get a free meal, which is good, and they like that, but we have them eat it in their cars. So that's sort of the in and out, and there was no exceptions. I mean, if you were the CEO of the whole place, you still went through the line. You still, had your temperature station. Everyone through the same area. So that was important. We also, if you go to a place of high infection rate, not for this plant in particular because it was out in the countryside, but for example, a partners group, if you go on their website, they have something called their pre-screen test, and they got 75 partners is the largest healthcare system in Massachusetts. I work with them also, and they every day 75,000 people come in and out of those hospitals and healthcare facilities, and we don't have any outbreaks. And the reason we don't is because there's an app that's other people can download too. If you're owning a business, you can actually download this app. We made it open to everybody. And you can actually have every worker check off whether they have any symptoms of COVID. And if they have symptoms, we say, go get a test and quarantine till you have till you have an answer for a period of time that's necessary. And usually that's about, usually we want to see no temperature for 3 days.
How many people are working at that particular plant?
At that plant, 300.
And that's both shifts?
That's both shifts, yeah.
Okay.
Yeah. So it gives you a sense. It was a mid-sized operation. It was a workstation operation. So a lot of the manufacturers have the additional challenge of, these guys are walking around, they're carrying heavy loads, they're having to type in to the same machine frequently as their partners. In those cases, the next thing we did after we got people into the plant was we tried to figure out which processes were the most dangerous. Which ones really require people to work together, work closely, work not closely, which ones could be removed. And for the ones that we couldn't socially distance, then we really had to figure out the PPE carefully. And so hopefully you're working with the groups that are going to watch your movements, are going to look at what you're doing, are going to try to help you with the right protective equipment all the time. Certainly everyone who was in those situations actually had a higher level mask. We got them an N95 mask, so just a normal cloth mask. We gave them frequently protective clothing and goggles, as well as face shields frequently, so that they really were protected. We also had those people who had to work in clusters stay in clusters. So we didn't let them, move around the plant a lot. We had workstation guys, who were on a shift. If you had 10 people on a shift on a particular workstation, we'd have those people stay together all the way through the day. And before the next group came in, we'd make sure we deep cleaned, so the next group of 10 cohort people would be able to come in and not see their other cohorts. Otherwise, you get both those groups sick. One cohort, they start to say, well, let me hand it off the shift. And all of a sudden you got all your best guys sick doing the same tasks, and then you got a big problem. So we try to actually spend about 20 minutes between the shifts to kind of let the whole place air down a little bit before we got the next shift in that had to do some of the critical tasks.
Now, how long have you been doing that at that particular facility? In other words, have we gone past the 14 days?
Oh yeah, we never shut down. We never shut down the facility. We were considered an essential good. In fact, we were ramping up operations, and we've gone from operations that were producing, say in the hundreds per day into the thousands per day now.
So, and has anyone gotten sick to your knowledge?
Yes, one person got COVID, and they had actually contacted 10 of their workers. Luckily, those workers were quarantined immediately for 14 days, and none of them actually came down with the disease, which was great. The sick person, luckily, seemed to have gotten it. These are tight, local communities who were all working together. so they were working together, and they were, barbecuing together, and they were from family, frequently from large families. And luckily, it didn't spread. So we had we started the process in March 1st, and we were planning it all the way from mid-February. So we planned everything, and it took a couple of weeks to plan everything. And then starting March, we implemented everything with all the new SOPs, all the new operations, and all the new laid out workstations. So we had to shut down for a little while to do that, and then we ramped back up again. And since for the entire period, we've had one person sick out of 300.
And was that in this country? Because I know you work with.
Yes.
Was in
Massachusetts and Rhode Island.
Okay.
Yeah.
All right.
Well, on a more broad basis, can you kind of give us an update on where we are with more universal testing and treatments and vaccines? All the stuff that, might give us some hope.
Well, yeah, there's always good reason to hope. So a couple things. First, Merck made a big announcement today. They are getting involved in a big way. They always were involved, but they've been sort of working under the radar. We're always sort of wondering. We've got 6 big vaccine producers in the world today. There's GSK and Takeda. GSK produces adjuvants, and they're going to give their adjuvants away, and they're working closely with Sanofi in a particular trial we can get into in a second. Then there's Takeda, and Takeda's decided not to work in this particular vaccine. But the other 4 are Johnson & Johnson, Sanofi, AstraZeneca, and Pfizer. And those are our big guys. So Merck is one of these big guys, right? And we were wondering where Merck was. Well, they bought a company in Austria that is working on a vaccine that they think they can put onto a vector that they've developed, an adenovector. They developed that was originally actually the measles and what they've done is they've taken the infective part of the measles out, and they've put now a new antigen in there, and that antigen is coming from this little Austrian company. And so that's one route they're trying to go after. And now the measles platform comes and infects and gets that antigen into the cell, then it's going to start to produce antibodies, and we'll get some immunity, hopefully. The second big group, it's not exactly the way it works. I'll talk about exactly how it works in a second. The second big thing is they announced they're going to reuse their Ebola platform, which is which is a recombinant RBS platform, and they're going to actually work with a company called IAVA, and IAV is a not-for-profit that has all these interesting pieces of antigen they can also insert into their Ebola vector. So that's the next piece. And then the last piece is they announced they're going to work with a company called Ridgeback to create an antiviral to actually start to impact the virus and hopefully cause it not to be able to reproduce effectively. So those are the 3 big areas that Merck just announced that they were getting into. Simultaneous to that almost was the announcement, of course, of Warp Speed. Now, Warp Speed is this operation that where President Trump has asked for 300,000 doses of a vaccine to be produced by the end of the year. And then anyone signing up for it has to agree that they're going to try to hit this goal, which is a really aggressive goal. And most recently, you heard about the Oxford Bioscience Group working hard on developing a vaccine. Well, AstraZeneca is going to come in on that with them, and they have signed up for Warp Speed, which is going to give them about $1.2 billion to actually try to create these new it's interesting. It's also an adenovirus, a measles construct that they're using with Oxford's special antigen.
Interesting.
So yeah, so there's a lot going on in the vaccine space, and we're gonna, I think, see some interesting results. We saw some initial results that we talked about briefly. We're going to start to see the important results coming out kind of in September timeframe. And the important results are we've gotten through phase 2, because phase 2, they actually have to decide what the dosing is and whether the vaccine is safe. Then you start to decide whether the vaccine is efficacious in phase 3. So those are but phase 2 is the one that really kills vaccines. Only about 11% of vaccines that are for novel viruses get through phase 2. Phase 3 is much easier, but it takes a lot more time.
So speaking of time, what are we looking at? Do you think before realistically we have a vaccine that we can distribute to the world? Another year from now, maybe?
Yeah, so that's a hard question. Usually vaccines take a long time to develop. I mean, I'm talking 20 years, 30 years. If you're on average.
I don't think Matt and I will be around that long.
On average, it takes 15 years. And in this case, people are trying to do it in 12 months. So it's a shocker. And the reason that we think we can do it are a couple reasons. First, well, some people think we can do it. I think it's going to be a little bit longer than 12 months. Unfortunately, I wish I could say it. I think I kind of did all the math, and the fastest I could get there was about 19 months from May. So it gives you a sense
From
of
May.
Yeah.
So that's the end of 2021 then.
Yeah.
Happy New Year 2022 then. Okay, gotcha.
Yeah,
All right.
and even that's extremely fast, and everything has to go perfectly. The people who are saying we think we can get there earlier are people who are looking at new kinds of vaccines that haven't been proven yet. And what these vaccines, so normally what we do is we take a little piece of the bug, usually it's the spike protein, and it's called truncated spike protein. That's what Sanofi's trying to do. And then they create the vaccine around that, and then your body says, it's a foreign object, and they create a defense against that foreign object. And so when you get the actual vaccine with the same spike protein, it's the same foreign object, and they go after it and they kill it. That's our immune system at work. So it trains the immune system. That's the idea of the vaccine. So that's one way of going after it, is just a piece of the protein. The second way you can do it is actually go after actually have the whole virus. And what they do is they actually attenuate the virus. So they break it down and make it. They kind of make it non-virulent, and they inject that in. Now the benefit of doing that is that you get a great response. And you only need one shot. And you don't need to have a very big dose, and you make sure the person doesn't get the disease by really killing the virus, make sure it doesn't work, and then you also give it adjuvant, so you stimulate your system also at the same time, and that creates the immune response. So those are the two basic ways that we have been doing things, either giving little pieces of stuff, little nanoparticles of it, or attenuating it and putting it in. Now, these other groups, they actually are injecting the RNA or DNA. And what that does is, instead of injecting the antigen or the piece of the virus itself into you, you're injecting the reproduction of a piece of the virus. So so then you inject it in, and the virus, you make the pieces of the virus. And then you also say, wait a minute, that's not right, and you kill it. And that's the way these new ones are, these RNA and DNA approaches. And so it's a different approach. And we've had nothing's ever been commercialized particularly with it. And by the third way, which I talked about, which was the sneaking in using a measles vector, or an RSV vector, or a horsepox vector, or some other vector. You put a little piece of the antigen on and you sneak it in that way. That also has a very good response, and also probably only require one dose. The thing about the RNA DNA is it requires 2 doses. It'll require at least, maybe even 3. It'll require what they call a prime. So you prime up the immune system, and then you boost it. What's nice about that is you can mix and match a little bit sometimes. So you can prime with one compound and boost with another. You have to be careful, but you can do that, so you don't have to have quite as much manufacturing capability up front. So there's some mixes and matches that you might be seeing coming out, depending on what we see these results as being. So it's an exciting time, and that's the reason that people think they can get down a little bit faster, is because they're using a new approach that's not as tricky to show that it's safe. Right, because if you're just injecting a piece of DNA, that's one thing. If you're injecting an entire spike protein or attenuated virus into somebody, that's a whole nother level of safety you got to think about.
Yeah. How about treatments? I mean, there's some viral diseases we still don't have a vaccine for HIV, but there are pretty effective treatments these days. So I mean, is there any hope for a treatment for this? Before we get the vaccine?
Yeah, in fact, we saw there's directly attacking the virus treatments, and then there are ways of attacking what the virus destroys treatments, right? So if you're getting ARDS, or if you're acute respiratory syndrome, or if you're having kidney failure, there are drugs for that. But now attacking the virus itself is what you're talking about, and there are kind of 2 approaches. The first approach is you try to give people antibodies, plasma antibodies, or now they're finding monoclonal antibodies, that kind of are in the system. You inject in, they're in the system, and they sort of glom up they sort of attach themselves and make the virus, very ineffective. It can't get into it can't get into your cells anymore because it's got so many proteins around it. And so that's one approach. Now, I've been working with some groups who are doing that work, and unfortunately, we're not seeing as many good, what they call neutralizing antibodies as we'd like to see. So this is sort of has been a difficult one so far. We've been actually going out to people and saying, you've been infected, you've survived, can we have some of your blood? And we'll spin that blood down. we'll look
we
for
want some of your blood.
You want some of your blood? Just a little bit. Actually, it's a pretty big bag. I shouldn't tell anybody. It's a pretty good size of blood that we want from you, and we spin it all down, and then we look for the antibodies, the IgG, IgA, and so on, antibodies that you've created as a result of those responses. And so far, we found about 3 or 4 really good ones that we think are neutralizing, and we're going to put those together. And now we're developing cells that actually produce these antibodies. Those are monoclonal antibodies, so we don't have to use the polyclonal antibodies, which we just use for prophylactic work and for very, very ill people. With monoclonals, you can produce a lot more of it, and you don't have as big an immune response as you would with a polyclonal antibody because it's just a one-shot deal. Then you can start to mix the antibodies also in interesting ways, but that's a ways off. That's probably. The plasma therapy is available right now, and if you have a ill person in the hospital who has access to plasma therapy, I would get it. If you're very, very ill, I think it can be helpful. And then let's see, 2 years down the road, we'll have these monoclonals. I think that'll be, and Sorrento's just announced 3 of them they think are very strong and so on. So they're seeing some of that. The other area you want to do is you actually want to stop the replication of, so what you can get in. The monoclonal antibodies didn't work. So now it's in, and you're trying to actually stop it from replicating. Those are the protease inhibitors that were so famous in treating HIV right now. As we then we let the virus in, we couldn't avoid it. So now it's in there. Are there drugs that can actually stop the replication of the virus once it's in you? And that's what we do with these protease inhibitors and so on. That's what remdesivir does. That's what the new Ridgeback deal with Merck does. That's what there's a cocktail now that's just come through and actually been approved by China, and we're going to put it on fast track in the United States. It's a cocktail of 3 drugs: beta interferon, lopinavir, and ritonavir combined with beta interferon. So those it's actually 3 drugs, even though we at level 4 because they've combined one of them already. But at any rate, the lopinavir is already been combined with ritonavir. Anyway, those 3 come together, and that's we're finding that remdesivir, we did the test in people who were quite ill. It was an open label test, and we found that people were actually able to survive slightly longer, not statistically significant, slightly more. So that's the most important number, but it wasn't statistically significant in the first round of the work. But we did find that it reduced the length of stay in the hospital from 14 days to 10 days. That's in the hospital, though. It's not just, that's it. So that was okay, but it wasn't great. What's interesting about the new cocktail is that actually is for moderately ill people who want to reduce their stays in the hospital. We think we can get that down almost 7 days for moderately ill, which is exciting because there's a lot more moderately ill than there are. There are about twice as many moderately ill as very, very ill in the hospital.
We got about 2 minutes left.
Oh,
So let's talk about those therapies now. Obviously, the vaccine is out of ways, but these therapies that may help minimize the damage, as it were.
Oh, yes. I didn't talk about those. Absolutely. So minimizing the damage, there are 2 big areas of damage. The first one is the ARDS, which is the acute respiratory syndrome issues. And those we're trying to, then you're hearing about, can we give them some heparin? Can we give them some TP? I'm sorry, that's not right. So there are 2 different things. One is the is when you've got thrombolytic issues, and there you got to talk about heparin and TPA. And the other is, are there ways of preventing the cytokine storm? And that's the ARDS issue. The cytokine storm, we're actually looking at a few things. One, things that block the cytokine directly. So there's IL-1 inhibitors, IL-6 inhibitors, one is from Novartis, IL-6 is from Roche, and so on, that actually block the individual interleukins. There's GM-CSF kinds of interventions as well. And that basically calms and resets your immune response so that you don't start eating yourself and immunizing yourself. And the other big thing, as I said, the thrombolytic effects. And they're talking about we're talking about things like TPA and heparin. And finally, we are getting some drugs for kidney failure as well, protect the kidneys. So there's some ways we can protect our organs in this event.
So good news in all of this, right?
There is. And by the way, I wanted to say that so that when you go back to work, we have 4 basic things you can do. One is distance. So you want to make sure you're apart from everybody. Don't sit across from somebody at a desk. That's the worst thing you can do. Make sure you're trying to get your place separated. Second big thing is hygiene. So make sure you're washing your hands every time you're talking with people. And that should probably be on average about 10 times a day.
Wow.
Yeah. Third, make sure that someone's wiping down your desk area and the high contact surface areas about once an hour. Good maintenance, and make sure that you're working in a facility that deep cleans every day. So those things, and so you've got the distance, you've got the hygiene. Then make sure you've got PPE. Make sure you're wearing a mask. Hopefully, your manufacturer will give you a mask. If they don't, then make yourself a mask. Make 2 of them and wash it every day, because after 24 hours it should be clean again, cleaned each time. So those are some of the things, and make sure that you're trying to get yourself tested before you get into, or every morning, do a check of the symptoms you've got. And if you have any symptoms, call in and say, I'm concerned I might have the COVID, and then hopefully they'll say, go get yourself tested. We should talk about testing next time, if you like, because it does take some time for the test to be effective. You can't just think you're exposed, you go right and get a test. It takes you about 5 days before you really should have your test done. Unfortunately, you have to sit that time out. If you think you've been exposed but aren't sure, the tests aren't accurate until you get about 4 days out.
All right, so testing next time, I think that'd be a great topic, because a lot of people want to get tested just to see if they have the antibodies.
Absolutely.
We're not allowed to do it unless we're showing symptoms. So it's kind of a circular thing here, so.
We'll talk about that in a lot next time, I promise. That'll be good. It'll be a good discussion.
All right. Thanks, Fred Brown, our epidemiologist extraordinaire. I can do it, so.
And it's fredbrown.com. Very simple.
Traditional 4-year students love Lawrence Technological University's thriving campus life, but LTU has always met non-traditional students' needs too. Lawrence Tech offers over 100 degree and certificate programs that can get adult students started or back on track. And most of our classes are conveniently offered evenings at our beautiful Southfield campus or online, so you can balance your social, family, and work life even while you power up your career. Lawrence Tech, where Blue Devils dare.